I'd like to join the rest of the speakers, and indeed the rest of you here, in expressing my appreciation to Jim as an advisor and a colleague, and in many ways a very exemplary model of what we would all like to be in so many aspects as scientists. And thinking about it, you sort of wonder, was Jim always that way, or is there hope for the rest of us? And was browsing recently through Jim's thesis from the University of Texas. I can't actually attest myself whether Jim was always that way, since I didn't know him really until I met him in '72 when I came to postdoc, but he probably knew me from the mid-40s. But if you look through his thesis, if I could have slides on, you find at the back of the thesis a CV, which tells you, if not whether Jim was that way, at least somewhat how he looked on himself at that time. And this was in the back of Jim's CV from his thesis in Texas. What I wanted to talk about in way of research today is actually in some way scientifically related to the first paper that Jim did after leaving Texas, which was on sex determination of flies. And as far as I know, the areas lay fallow in his lab until I came there in the mid-70s and took up and started working on sex determination, which has been the major aspect of what my lab's been doing since then. And in terms of sex determination, what I'm going to talk about is where our knowledge stands today, and most of it comes from looking at what determines sex in terms of the cuticular phenotypes on a fly. Meaning, here we have a male and female abdomen. You can see the pigmentation of the male differs from that of a female, as does the external genitalia. And similarly, on the foreleg of a male, we have a sex cone versus a homologous row of bristles, which are different in number and structure in the female. And most of what I'll say about working out the regulatory hierarchy controlling sex determination is done in terms of how it affects these external cuticular aspects of a fly, go from what we know about the internal biochemistry, the behavioral differences between males and females. What we've deduced from studying the cuticle applies to all aspects of somatic sex in a fly. And to give you an overview of where we're going, I have here the hierarchy, the regulatory hierarchy as we know it, that controls sex determination in flies. The initial determination being the X-autosome ratio. With equal numbers of X's and autosomes, one becomes a female somatically, and in the germline it has a female level of X transcription. Whereas if there's one X and two sets of autosomes, one becomes a male in terms of somatic and germline sex, and the X chromosome is hypertranscribed to provide for dosage compensation. There's one gene we know of called sex lethal that's primarily been worked on by Tom Klein in Princeton that seems to control, be downstream from the X autosome ratio and control both somatic sex and dosage compensation. But the regulatory hierarchy that we know downstream from that diverges in that there's a set of male-specific lethal genes that are on in a male and responsible for causing dosage compensation. These genes are not expressed in the female and do not affect sex determination, whereas there's a set of genes, double sex transformer, transformer 2, and intersex, whose states of activity in male and female somatic cells are responsible for determining all aspects of somatic sex. And the part of this regulatory hierarchy that I want to focus on today is the aspect that controls somatic sexual differentiation. One of the first questions you might ask about these sets of genes are whether they determine sex in an individual cell independent of what the rest of the fly is, or whether they make diffusible substances, such as hormones, that are governing sex. And one of the experiments I did while postdoc in Jim's lab was to construct mosaics which had patches of tissue that were homozygous for one or the other of these mutants and affected sex determination, and ask whether those patches of tissue showed the phenotype of the mutant or the phenotype of the surrounding cells, which were wild type. And if I could have some lights on, the phenotypes of these mutants that we'll be talking about, if I don't get tangled and stumble, are mutants transformer and transformer 2 affect sex in just female individuals and cause female individuals to develop as males. in all aspects somatically, in terms of how they look, in terms of how they behave, in terms of their internal biochemistry. And these genes have no, mutants in these genes have no effect on sex determination in males. Whereas the gene double sex, when you knock it out, causes both females and males to develop into intersexes. And similarly, the mutant intersex causes females to develop into intersexes, but doesn't have any effect on males. When you construct the mosaics I talked about, in which we have a patch of tissue in a female, say, that's homozygous for a transformer, in an otherwise wild-type female, what you see is a result like this, where we have a wild-type female abdomen in which we have a patch of cells homozygous for transformer that cause those cells to have a male phenotype in terms of pigmentation, and this, so that this gene is acting autonomously to determine sex of individual cells. And the same result is true for the other genes in this hierarchy. So their sex seems to be determined completely autonomously and not by the physical substances, at least as we know the genes. In terms of having a set of regulatory genes like this, one of the things one would like to know is how do they interact with each other to bring about sex determination? What is the structure of the regulatory hierarchy that they're involved in? And to do that, what we did was to define the null phenotype of these genes by finding deficiencies for them, and using a knowledge of what a complete knockout of the gene was, together with a phenotype of double mutants between these various genes, construct a regulatory hierarchy from those interactions. And that is shown here. And I'll go briefly through it and then just a little bit of the data suggesting that led us to this sort of model. The double sex gene is, at least the time we did this, unusual for a eukaryotic gene, in that it seems to have two different active functions, one of which is expressed in females, the other of which is expressed in males. If a double sex gene finds itself in a chromosomally female individual, it's expressed in what we refer to as the double sex female mode, and that active function is to repress male differentiation. Female differentiation is not repressed, and so that individual develops as a female. Conversely, if the double sex gene finds itself in a chromosomally male individual, it expresses the double sex male function, and the double sex male function is to repress female differentiation. Male differentiation is not repressed, so that one gets a male development. The control as to which of these two states double sex is expressed in appears to be via the transformer and transformer 2 genes, which are turned on in a female and have as their active function causing double sex to be expressed in the female mode, repress male differentiation, and therefore allow female differentiation. In a male, transformer and transformer 2 are not expressed, and so double sex stays in what we think of as this ground state, the state of expression characteristic of a male, in which it represses female differentiation. And to just show you one of the bits of data that suggests that this structure to the regulatory hierarchy, in particular that double sex has opposite negative functions, I'll just tell you a couple of pieces of data. One is that null mutants at double sex knock out both of those functions and convert both males and females to intersexes. As one might expect, if you could repress neither kind of differentiation, then both would occur. And in fact, that's the phenotype of those flies. They look as if both pathways of sexual differentiation are being followed simultaneously by cells. And one of the ways one can see that most dramatically is to look in the derivatives of the fly, of the genital disc in the fly, which give rise to the external genitalia and the internal ducts. Because in each fly in wild type, there are two primordia, similar to the situation in mammals. It's a set of primordial male and female cells. In a wild type male, the male primordia divides and develops and gives rise to male genitalia and the internal ducts, whereas the female primordia doesn't give rise to any structures in the adult, it's repressed. And conversely, in a female, there are again the two primordia, but in this case it's the primordial cells for the female genitalia which develop into the genitalia and internal ducts, and the cells in the male primordia don't develop at all. If, on the other hand, one looks at what happens in individuals that carry a complete loss of function at double sex, one finds that regardless of their chromosomal type, one gets both male and female genitalia produced from the two primordia, so that both kinds of differentiation are simultaneously occurring. And if you look at what this means logically between comparing what the loss of double sex versus the wild type function must mean in a male, that is, looking at the difference here, we see when we lose double sex function, we allow female genitalia to develop, so female differentiation is occurring, so that the wild type function must be to prevent that or repress female differentiation in a male. Conversely, if we look in a female, we see the difference between wild type and complete loss of function is the development of male genitalia, so that the wild type function of double sex in a female must be to prevent male differentiation, so that it has opposite negative functions in the two sexes. And one of the interests in this, being having two different opposite functions, was that, and controlling sex, was that this gene then has substantial parallels to the mating type locus in yeast. So that to begin molecular work on this hierarchy, we started out by focusing on double sex. And to do that, we took a dominant mutant of the gene, reverted that with X-rays to collect a series of translocation and inversions, all of which were broken in this particular salivary band, which told us where that gene was. We also collected other rearrangements around the region, including a deletion that deleted every gene up to double sex that didn't affect double sex function, so that provides an outside bounds on the gene here, and a translocation broken in a flanking lethal complementation group on this side that provided a flanking bound there. In addition, we obtained a deficiency, one of whose endpoints was within the gene, the other whose endpoint was just short of a tubulin gene implies. That tubulin gene had been cloned in Meredith Pardue's lab, so a postdoc of mine, Mariana Wolfger, and I took DNA from that tubulin gene, walked out till we found this breakpoint, then cloned the fusion fragment from here into double sex, and then got off with the DNA from this end, walked off through the double sex gene to identify where it was by the breakpoints we'd induced. And we found the translocation and deficiency, which provided outside bounds of the gene, about 75 kilobases apart, and the inversions and translocations that were broken within double sex itself by the fact that they inactivated the function, spread across a region of about 30 kb in the middle of that. Given the formal genetic parallels between double sex and the mating type in yeast, one of the first things we did was to ask, were DNA rearrangements responsible for the difference between male and female expression of the double sex locus, which we did by looking to see if there was any difference in restriction digest patterns when we compared DNA from adult males and females. And the results of that were that there were no differences between males and females, so the regulation had to be at another level. So we began to look at transcripts across the region to see if there was any difference there. When you look at the transcripts across the region, where here is the region in black, which is expanded here, in which breakpoints occur that inactivate the double sex locus. If one takes various fragments of that and probes northern blots of RNA collected from various developmental stages in males and females, one sees, depending on the probe, different patterns of transcripts. With some particular probes, one sees transcripts which are present in both males and females in larvae. However, as one goes to the pupal period and uses different probes, one can see transcripts here present in a female versus several that are present in pupal males of different sizes. Here's a transcript that's present in adult males but not in adult females. So that there's substantial differences between the sex in the kind of transcripts that one sees from the gene. And this is just using some different probes from across the region, showing that one can again see sex-specific transcripts as well as transcripts which are present in the larvae that are not sex-specific. This is summarized here, where this is the pattern of transcripts we would see in larvae, pupae, and adults of the two sexes, so that there are larval transcripts that are sex non-specific, and then pupal and adult transcripts that are sex-specific. And where those come from in the gene is indicated by the open boxes here. So that these transcripts are formed by a variety of uses of the DNA from the double-sex gene to generate those different transcripts. I can say from analysis of cDNAs here that these differences are due at least to 3 different splices that we've identified, and also to 2 different polyadenylation sites that we've identified on these transcripts. And quite possibly, but we haven't proved it yet, this may also have a different 5' end than the rest of the transcripts. So that we have in double sex then, with the fact that there are sex-specific transcripts, a molecular basis for the different functions, at least in principle, that we need to explain the genetics, but obviously we've got far too many transcripts and we need genetic functions. The genetics only needs a male and female function, whereas we have many more than 2 different products from the gene in terms of transcript analysis. We have done a number of other things with double sex, a couple of things at the DNA level. One is to ask whether this regulatory gene, that's obviously important in melanogaster for sex determination, is also present in other species, so that we've looked across various Drosophila species and some other Diptera, and it's not surprisingly found throughout various Drosophila species that we've looked at, but most interestingly, it's found in the housefly Musca domestica. And that's of interest because in the housefly, the primary sex determination mechanism is like in mammals. It's a dominant Y chromosome that determines sex. You've got the Y, you're male, unlike the situation in melanogaster. And yet we can see good homology to probes from double sex there, suggesting that this regulatory hierarchy is conserved quite broad and probably then quite ancient in its evolutionary origin, and may be used downstream from a variety of different primary sex determination mechanisms. We also, at the DNA level, have sequenced one of the cDNAs. It's about 2.5 kb, corresponding to one of these transcripts, to, at the first level, put it into a protein database and ask, is it anything known? And I can tell you that it's not an oncogene, it's not a growth hormone receptor, it doesn't have a homeo box, and I think most fortuitously it's not some basic metabolic enzymes. I prefer not to get up here and tell you that the basis of sex was, say, 2 different forms of G6PD. So the inferred amino acid sequence from the gene isn't telling us anything particular at the present moment. We've also asked something about the functioning of this gene, and that's in the following sense. If you look at a fly, you see a small fraction of tissues are, to our eyes, sexually dimorphic. The pigmentation patterns, we know the fat bodies are dimorphic and the females make yolk proteins and males don't. There must be central nervous system differences because of behavioral differences that are under the control of this hierarchy. And so one could imagine that it's the case that only some cells in the fly know their sex, and for the rest of the cells, their sex is irrelevant. Or it could be that we just can't look at enough detail, and then in fact this hierarchy is functioning in all of the cells in the fly. And those have 2 different implications from a developmental standpoint. If all cells know their sex, then it's easy to think of the functioning of this hierarchy as reasonably autonomous from the functioning of the various hierarchies that control segmentation in flies. They are at least controlling the expression of these genes. They may interact, their products may interact together with the products of these genes to control differentiation functions, but they're not talking directly to each other. On the other hand, if this hierarchy is expressed in only some cells and not others, then there must be information in the cells which are controlling the expression of these genes and dictating where they're expressed or not. So to do that, what we did was to do a hybridization to tissue sections to look at which tissues in a fly express double sex, and therefore and whether all cells or only some cells knew their sex. And the results of that are in fact a mixture of the 2 hypotheses that I told you. There's one tissue in a fly which knows its sex very well compared to other cells, in that it has a lot of transcript compared to other cells, but all cells seem to know their sex. This is a dark field section through a pupae in which the silver grains are here. This is an imaginal disc and a variety of other cell types in the pupae. That's the phased section. I think I got these in backwards. This is the tissue that knows its sex much better than all others. It's the proventriculus. It's part of cells around an anterior part of the gut. And in the bright field here, you can see all the cells in the background that didn't light up at all, whereas this little patch of cells here does. But if you took a longer exposure, you'd in fact see hybridization for all the tissues there. So, and I can't tell you anything about why this part of the fly knows its sex much better than other parts, and nobody I've been able to talk to has been able to suggest any function there that's particularly special. So that it seems that this hierarchy is, in general at least, expressed in all different cell types. Having done that with double sex, and the work there is obviously still ongoing, we set out to look at the other genes, transformer and transformer 2, that we think control it at a molecular level. And John Belode and Mike McEwen in the lab, together with Mike Hoffman, who's here, carried out a saturation of the region around the transformer in terms of finding a variety of breakpoints and defining a set of complementation groups in that region, and took clones from the scarlet gene and from homologous to an Abelson sequence at this end and walked across several hundred kb, identifying all the breakpoints along the way, and we're able to define transformer as being between this deficiency breakpoint, which doesn't delete it, and this deficiency, which does delete it. To find out where transformer was in there, they first looked at the mutants that were available, and the first mutant that the mutant used to define the locus was found by Sturtevant in 45, and that chromosome has associated with it a 1.2 kb deletion at this point. There's obviously no parental chromosome for that, but that's at least a possible site for the transformer gene. They then looked at transcripts from across this region, and there are a variety of transcripts, as you can see. It's a very heavily transcribed region, but one of those is transcribed in a female-specific manner to make an RNA of about 1.1 kb, and that comes from exactly the same place as the transformer mutant has a deletion of 1.2 kb. So together that correlates very well with what we know genetically, which is that transformer seems to be expressed just in females and not in males. To obtain a definitive proof for that, Mike and John cloned the region around here, put 1.8 kb of that into a p-vector, reinserted that into flies via transformation, and were able to show that that piece of DNA they reinserted could, in some sites at least, completely rescue the phenotype caused by the transformer mutant. So that is the transformer gene. And is, compared to most homeotic genes in flies, a nicely small one. The maximum size of the gene can only be about 1.5 kb because of the closeness of the flanking transcripts. So it should be reasonably easy to analyze. What I want to turn to now, in the last few minutes, is how this hierarchy functions to control the genes downstream from it. That is what we've talked up to now about the regulatory genes, but they're all there to control a set of genes involved in sexual differentiation. And one of the most fruitful approaches we've taken to that is to use the transformer 2 gene, in which we have several temperature-sensitive mutants, to use temperature shifts with those mutants to define when and how this hierarchy is functioning to control expression of the genes involved in differentiation. One of the first sets of shifts we did was to look at various cuticle phenotypes and found, much to our surprise, a result which we hadn't expected. That is, we'd expected that if you did look at a particular kind of cell, you'd find that in that cell, at least, this hierarchy was functioning at one time to determine its sex. But instead what we found was that within one particular cell lineage, the hierarchy was in fact functioning at several different times to control different aspects of sex in the same cell, as if it was needed in some way to make a decision and to perhaps maintain that decision or to make a series of sequential decisions. Here you see the results of raising flies carrying a temperature-sensitive transformer 2 mutant at a permissive temperature where they develop as females. And here's the female row of bristles in the foreleg, which has the female number of bristles and the female morphology, raised under restrictive conditions, the male morphology and the higher male number. And with appropriate temperature shifts, you can develop forelegs that have the male number and the female morphology, or the female number and the male morphology. And it's worth noting that each of these bristles is the product of a single cell. So within a single, there's separate decisions as to how many cells will produce bristles, and if they do produce a bristle, what kind of bristle they're produced. to look in a more specific way at how this hierarchy controls sexual differentiation, we focused on 3 particular proteins that are under the control of this hierarchy, namely the yolk proteins, which are made in females in the fat body and the follicular cells of the ovary. And these 3 yolk proteins are the major storage proteins of the egg. And we know from previous work that they are under the control of this regulatory hierarchy, that if you transform a female into a male with a transformer mutant, the yolk proteins aren't made, whereas in a normal female they are made. So one can do temperature shifts and look at the synthesis of the yolk proteins, which are here. If you raise a fly at the restrictive temperature, the yolk proteins are not made, so that's an individual reared at 29 degrees develops as a male, doesn't make them. Whereas a wild-type female makes substantial amounts. If you shift that individual from 29 to 16 degrees as an adult, then very quickly, within a day, and in fact another experiment, 6 hours, you can see synthesis of those proteins starting and reaching wild-type levels. So that this hierarchy is needed to turn on the expression in the adult of the yolk protein genes. And with the reciprocal temperature shifts, where we raise individuals to adulthood as females, in which case they are making yolk proteins, and then put them at the restrictive temperature for various periods of time, one sees the amount of yolk protein synthesizes decreases, saying that the hierarchy is needed not only to turn on these genes, but to maintain their expression. And if one uses probes for the yolk protein genes which have been cloned and were given to us by Peter Wensick and looks at transcription, one can see that this control is at the transcriptional level. And so within a few hours, one can turn on or turn off transcription for the yolk protein genes in the adult. So that this hierarchy not only is functioning during the larval and pupal stages to specify development, but is actively used in the adult to maintain at least one aspect of sexual development. There's one other aspect of sexual development that we've looked at in the adult, or sexual differentiation, and that is behavior. I've alluded to the fact that these genes, transformer and transformer 2, not only transform cuticle flies with respect to their cuticle phenotype, but also with respect to their behavioral phenotype. A female homozygous for a transformer mutant behaves like a perfectly good male in that it carries out with good fidelity all of the male courtship patterns, which are an innate behavior pattern that's present in adults. And so we took adults and, well, we did 2 things. We raised individuals at either the permissive temperature, in which case they develop as females, to show almost no male behavior, where we assay male behavior by following pairs of flies, assaying whether they orient or not to each other, carry out wing vibration, whether the male licks the female genitalia, attempts copulation, and succeeds. Raised under permissive conditions to adulthood, these individuals don't show male behavior. We raise them, on the other hand, at the restrictive condition, then a substantial proportion of them show good male courtship behavior. Not all succeed in achieving copulation, but because of the genotype we used, these individuals don't have perfectly good genitalia, and so that may have been mechanical reasons why they didn't all succeed in actual achieving copulation. If you take individuals that have been raised to adulthood as females and put them as adults at the restrictive temperature for about a week, one finds that about a third of those now show various amounts of male courtship behavior. And so it must be that 2 things. One is that this hierarchy is functioning in the central nervous system of the adult, since we can shift these flies and change their behavior, and we know that that behavior from work in Jeff Hall and Seymour Benzer's labs maps to the central nervous system, not the peripheral nervous system. So that hierarchy must be functioning there, which must mean that rather than flies being hardwired, as you normally think of insects being in terms of their central nervous system and innate behavior patterns, they must be in some way plastic, at least at a functional level, and that plasticity or the particular state of differentiation that you see in an individual is something that's maintained actively by controlling the pattern of genes involved in the differentiation being regulated in the adult. So that in this aspect of sexual behavior, as in the case of the yolk proteins, we have substantial regulation taking place in the adult in terms of maintaining particular states of differentiation, as well as largely in pupae, where we have taken place the overt differentiation we see in terms of the cuticle in the fly. And just to close, I'll say we have very recently in the lab cloned the transformer 2 gene, which is what we've used to carry out these temperature shifts with. And so we can now begin to use that and look at its expression and hopefully use it in ways which will let us get directly at some of the genes involved in controlling behavior at other downstream functions. I'll stop at that point.